Every day, researchers and clinicians are changing the world. They are developing new treatments, making scientific discoveries, solving complex problems, and asking questions that could shape the future of medicine, health, technology, and society. But most people never hear of their stories. Welcome to Dr. What, a science communications podcast exploring the people, stories, and ideas behind research. Each episode, we sit down with experts from across science, medicine, healthcare, and innovation to talk about their discoveries, career journeys, biggest challenges, and what the future might hold. We will explore everything from cancer and mental health to infectious diseases, climate and health, artificial intelligence, and groundbreaking discoveries that could transform our lives. And because science belongs to everyone, we will also be asking our guests questions from the public, sometimes insightful, sometimes surprising, and occasionally completely unexpected. Welcome to Dr. Watt. The doctor will see you now. I have with me today Professor Jeremy Green.
Hello.
Yes.
Just to introduce myself. So I'm professor. My official title is Professor of Developmental Biology
Okay.
at King's College London. And what I do is to really just ask the basic question that I think many people have when they see something in nature is, how did it get to be there?
Yeah.
And in my case, I'm very interested in the sort of forms and shapes of things, in particular animals, how you go from a single cell, we all start as single cells, to something with head, arms, legs, muscle, bone, blood.
Yeah.
And as a professor at King's, I do a mixture of research. So I have a research group, a team, as well as doing teaching. And I teach everything from the most basic biochemistry to my own specialist areas,
Yeah.
describing, again, stuff that was done 150 years ago and stuff that we did last week.
That's amazing. It's just incredible how your journey has been. Because my first degree was in biochemistry, but I don't think I even know what my trajectory is going to be in the next 5
Actually,
years.
that was my degree. My
Was
degree
it?
was actually in biochemistry. But
Yeah, so let's go back a bit to
okay.
how this all started for you. How did you get into this field?
Sure. So I as a youngster, and I think most academics start out being pretty nerdy as
Yeah.
kids, and I was sort of interested in everything. And I thought, well, you can be a scientist who knows a bit about non-science, but it's much harder to be a non-scientist and know about science. So I
Yes.
thought I better do science so I can know more.
Yeah.
Um, and neither of my parents was a scientist, although they were quite admirers of science. So I did science. I did an undergraduate degree. I was at Cambridge, did natural sciences. And I wasn't particularly focused on biology, so I did maths, physics, chemistry, and then a wonderful course that they did there called Biology of Cells, which was basically to seduce people from what you might call the dry sciences into
Yeah.
biology, and it worked in my case. And then I did, chemistry, biochemistry, history and philosophy of science. In my third year, I did my other courses, biochemistry primarily, but one of the modules was in developmental biology. And developmental biology at that time at Cambridge, as it still is, and as it is actually generally in the UK, absolutely top class. So one of the people who I attended the lectures of and later came to know personally was John Gurdon,
Yeah.
recently deceased, Sir John Gurdon, who got the Nobel Prize a few years ago. And I just loved that course. And when I was trying to decide what to do after graduating, I didn't really know what I wanted to do, but one of the options was to do a PhD. Considered other things.
Yeah.
But I thought, if I'm going to do a PhD, what do I want to do a PhD in? And science is huge. And at that time, we didn't have the internet, but there was a catalog, paper catalog of PhD places available that year. And there must have been probably a couple of hundred. And so I had to think really hard, what is it about biology that's really cool. I mean, really, what really motivates one's wonder in biology. And for me, it was just trying to understand how you could organize structure, how things could get organized. How would you make a hand? You look at your hand and you go, well, where does it begin? I mean, how do you begin
Yeah.
to make that when you don't have a plan or you're not a sculptor? I mean, you look at the religions and, some god is usually shaping something, but of
Yeah.
course biology doesn't do that. It's got to do it itself. So how
Yeah.
does it organize itself? And that question has really sort of stayed with me, and we still work on it. So I ended up actually doing a PhD in something slightly different. But then after finishing the PhD, which was in actually the gene regulation,
Yeah.
in particular gene regulation in yeast. So actually not at all related, single cell organism yeast.
Yeah.
And it was in fermentation, so looking at the enzymes that enable yeast to make alcohol. But looking at it at the molecular level and doing molecular biology, which is, if you like, the technical bedrock of everything we do in biology these days. And so I was looking at gene regulation, which is involved in everything in biology. But then for my postdoc, I joined the lab of Jim Smith, now Sir Jim Smith. So I don't just work for people who are going to be knighted. Seems to be a theme here. But anyway, at that time, Jim was a starting investigator, and I was not his first, but his first sort of persistent postdoc, if you like.
Yeah.
And he, at that time, had revisited a puzzle that had been in the field for a number of years. And the story goes back, there's a bit of history here, sort of history of science, which is probably worth recounting. So back in the late 19th century, actually late 19th century, early 20th century, people were trying to understand how embryos were made, and they mostly were doing experiments with various marine species, but also with frogs, because frogs lay eggs. You see the frog spawn in the pool. You have a jam jar of frog spawn and watch the tadpoles develop. And because they're very big and accessible, unlike mammals, they don't develop inside the mother. You could do experiments on them and sort of see what happens. And they just grow in simple pond water. It's so easy to work with. And probably the most famous experiment of that era was called the Schmemann organizer experiment, where actually a PhD student, not the man himself, although he, I guess, directed it, but took and it was a female PhD student, pretty unusual at the time, in the late 19-teens, took a graft from a particular anatomical region of the early embryo, which is just a little dimple on the surface of those black dots that you see in the frog spawn. And she took it and grafted it onto the opposite side of a pale-colored other. As it happened, they're working with newts, not frogs, but it was another amphibian. Yeah. And they ended up with these tadpoles with two body axes.
Oh, wow.
So you had these sort of monsters that had two spinal cords, two brains, two pairs of eyes, and so on and so on. But the key thing was that they could trace where the tissues came from, and they could see that a lot of the second body axis that was generated or provoked, if you like, by the graft came from the host. In other words, it was not part of the graft that was just growing well in a different place. It was actually sending signals. And that was the first realization that in biology, if you like, things signal to one another. They send chemical messages between them, different tissues and cells, that can instruct cells to do something different from what they would otherwise do. And that immediately after that, there was a period where people were trying to find out what those substances were. And they just did not have the technology, the biochemistry actually, to identify those things. And that whole agenda kind of lapsed. And part of the problem was there were lots of, not only because the technology was not adequate, but also because it was very easy to get a false positive, as it is generally in biology, because biology is generally self-organizing and does a lot of stuff sort of on its own, which is part of the question. But it's very hard to stop it doing that and making sure things are under control. So that whole hunt for those substances was a sort of considered a very risky area. People avoided it. And then in the mid-80s, Jim Smith, uh, tested out a particular a medium in which frog cells had been growing. And he found that one of the activities of this medium was to trigger formation of what in the embryo is called mesoderm, which is basically the precursor of a whole series of tissues like muscle and bone and blood and the kidneys. And I came along to his lab, and my head was filled with the theory that I'd learned as an undergraduate.
Yeah.
And one of the big theories then was this thing called positional information. And the idea was, and this is a big idea in developmental biology, that you can organize a whole pattern by having a chemical gradient. So if the cells, which can all be the same to start with, see a high amount of that, they'll do one thing. If they see a middle amount, they'll do another. And if they see none or a low amount, they'll do a third thing.
Yeah.
And that way you can imagine organizing a pattern. And people who may have done a course in this would be familiar with this thing as being called either positional information or the French flag model, because the kind of icon of this is a tricolor flag.
Yeah.
So I came to the lab and said, well, I wonder if this inducing factor that Jim had found was actually a morphogen. That's a word that is used to describe that chemical. I wonder if it's a morphogen. How do you test whether it's a morphogen or not? And the answer is you give different doses to cells and ask if they become different things. And it wasn't a completely easy experiment because cells don't normally live individually. They live in tissues. So how do you make sure they get the right dose and all of that complexity? But without going into the technical details, I was able to do that experiment and show that this particular ingredient in this medium fitted that model perfectly. So at high doses, it gave one thing. At middle doses, it gave another. Actually, it gave 5 different things depending on the dose, and they were separated by very sharp dose thresholds, basically. So anyway, that was my entry into developmental biology as a postdoc, and it's sort of my claim to fame in the field, in the sense that that really, for the first time, identified the molecules that do that job. And we now, 30-something years later, have a sort of a palette of those ingredients that is the basis for being able to direct cells to make things like organoids. These are sort of mini bits of tissue that you can test drugs on and so on, or that mimic particular diseases. And also the idea that you can use stem cells to regenerate people and repair organs and so on. So that was the foundation of it. There are other people working on related things in parallel, but that was the first demonstration that these were the real morphogens that up to that point were almost entirely theoretical.
Yeah, that's amazing. That's amazing. Wow, thank you. It's just amazing how starting just in biochemistry, leading all that journey, just going through the discovery of morphogens. And what do you think are the elements that drive discovery and innovation, like in your field, and which can be applied to other fields of science, like cancer biology? Or
I mean, I think you have to think of this from a bunch of different perspectives. So I think I'm sort of at the personal level. I think one of the things that people who haven't been there maybe are not quite so conscious of is that science is really hard, in the sense that if you're working at the cutting edge, you at some level you don't know what you're doing. I mean, if you knew what the answers were, you wouldn't be doing it. It wouldn't be research, right? So there's a lot of trial and error, and quite obviously that means there's a lot of error, which means a lot of time one spends in the lab or in the research environment, whatever it is. There's a lot of negative. There's a lot of failure. That's normal. That's one of the things you learn, actually, even as a PhD student, is you have to be resilient. You have to be able to take that failure and just treat it as the norm.
Yeah.
I mean, obviously, it's not all failure, right? But depending on the field and a bit of luck and so on, that's the reality of your daily life is trying to figure out how to do something which nobody's done before, is kind of maybe quite difficult or finicky
Yeah.
to get done. Making it work, doing an experiment and finding that the result is negative or the experiment didn't work in the way that you could explain or predict or had hoped would work.
Yeah.
So you have to do it again and again. So to be in science, you really have to be motivated. And so for me, it's that I just find the whole, well, I like problem solving. I like puzzles. That probably helps. But you also have to be fascinated by what you're looking at. Because if you're not really fascinated in it, it's just too hard. It's just too hard. You really have to have that motivation. And I still, I do occasionally go into the lab and do some practical work, although a lot of the time I'm at my desk trying to write grants and get money. We can talk about that later. But I still look at this progress from being an egg, a fertilized egg. It divides into multiple cells.
Yeah.
And 2 days later, you've got a tadpole, you've got this animal in the dish. We still work with frogs. I do quite a lot of stuff with mouse now. But occasionally we do frog experiments. And you just look at that and you think this thing with the eyes, and they sort of tremor a little bit in their sockets, and the heart's beating and the tail is twitching. And it's just this, and it's beautiful. I mean, it's just amazing. It's just an amazing thing. You used that word before. I mean, biology, nature is amazing, right?
Yeah.
How can one not be fascinated? And you look at the human body, you got fingerprints, right? How do they get to be there, right? Or the nail, or why is it this texture? Why are nails hard and skin soft? Why is it that stuff that we encounter every day? And if you just take a moment, you go, wow, how does that be? And knowing some people say that sort of reducing it to science and molecules is somehow unweaving the rainbow. I can't remember who said that, but it's actually, I think that knowing how things work as well as being able to sort of behold them increases one's awe at the whole thing rather than decreases it. So I think at the personal level, at the subjective level, that's the answer to the question.
Yeah.
There are also things about policy and how you manage and organize the whole process that are also important for innovation and progress. And I sort of lean on the side of, but I think this is an important general point. I kind of lean on the side of recognizing that when we have heroes in science, Nobel Prize winners and so on, or stars, I think that's it's always post hoc. It's always a rationalization. We like stories. We like heroes. We need heroes. I mean, I have heroes too. But actually, science in reality, everybody who does science knows that in reality, those Nobel Prize winners are not that much more brilliant than anyone else who's at that level.
Yeah.
I mean, obviously, you have to have a certain level, but they're usually the end point of a process involving immediately a handful and almost always dozens of predecessors and colleagues, and who happens to be in the right place at the right time and have the right persistence and market themselves and all of that stuff. They're the people who get the prizes, and we make them heroes. As I said, we need them. But actually, the best way to get good science done is to let, to buy a lot of lottery tickets.
Yeah.
But they have to be gold. They all have to be golden tickets. But you need a lot of them. So it's better to. I think science is fundamentally quite a democratic process.
Yeah.
Doesn't matter how elevated you are. And again, if you're one of these quote unquote superstars, you still have to submit to peer review, and that's good. That's healthy.
Yeah.
There shouldn't be. Authority is always a mistake in science, and one of my favorite things is the motto of the Royal Society is nullis in verba, which means don't take anyone's word for it, basically.
Yeah.
And that applies even to the most elevated scientists. So that's kind of the two levels where I think innovation and advances really happen.
It's amazing, and it's quite interesting that you talk about science having a lot of heroes, and that is really quite a bit of element in Doctor What podcast, because a lot of people also see you as a hero. So if you were going to be if you were a superhero, who would you describe as your sidekicks? The people that are behind the scenes and make it happen. Your Robin Hood. Your the
Oh
second, yeah,
well, I mean, everybody who's a professional scientist is working these days in a team, and I mean, there may be some fields where you can do things as an individual, but by and large, certainly in biology, it's uh, if you're doing anything experimental, you need the whole team, and that's everybody from and I don't even want to put it in a hierarchical
yeah,
way, but it's everybody from a very experienced postdoc who's looking for a faculty position, a permanent position, a lecturer or assistant professor, whatever it is, the most experienced to the newcomer master's project student.
yeah,
I mean, they contribute too. I mean, I've had papers that have emerged directly. In fact, whole programs that have emerged directly out of something that an undergraduate did, even.
yeah,
So I remember many years ago. So part of the things, one of the things we look at how it gets made is teeth. So
yeah,
I happened to be sort of coincidentally, but quite valuably in many ways, and with and it's a great benefit for me is I'm based in the faculty of dentistry and oral and craniofacial sciences at King's,
yeah,
FODOCS, and so a lot of what I focus on is development of things in the head. So the anatomy of the head is very complex and intricate, and there are lots of things to look at, many things. About half of the embryo is head, actually.
yeah,
So it's a sort of a big place, both literally as well as metaphorically at certain stages. So one of the things we look at is teeth. How do teeth form?
yeah,
How do they make themselves? How do they begin? And it's a kind of a cool thing which I'm happy to describe. But um, I didn't start working on teeth until I had an undergraduate who was doing an intercalated degree,
yeah,
who said, "I want to be a dentist. Can I look at teeth?" And I had a fairly clear idea about how I thought teeth developed. And in his project, he did the work. And although I have to say it wasn't that great a project, but nonetheless, I mean, he didn't do it that well. But the fact is that he did enough to say, well, actually, maybe the way we think teeth are made is not correct.
yeah,
And then I thought, well, actually, maybe I should write a grant application, get some money to look into this properly. And I did, and we did. And we now know how we get to about 2/3 of the way through development. And I'm trying to get the next grant
yeah.
to look at the last bit, basically.
Oh, that's amazing.
Uh,
And also with the element of working in a team, you're also the vice dean for international FODOCS as well. So what do you think the role of teams, collaborations, interdisciplinary research has to play in driving discovery and innovation in your
Well,
field?
so, I mean, of course, you need disciplines and you need to have specialist knowledge, but actually, it's fun and motivating to work with other kinds of specialists,
Yeah,
and they can bring things to a project that otherwise you couldn't do.
yeah,
So in my case, I mean, partly because of the stuff I did as an undergraduate, and partly it's just the way I think. I kind of like the physics of biology. How do physically things get made? What are the mechanics? So a big part of biology in the last 30 years has been almost obsessively focused on genes, gene expression, genetics,
yeah,
genomics, the Human Genome Project, all of that. And that's amazing and important and vital, and of course has ongoingly. But genes don't make things. Ultimately, most of what we see around us in bodies and tissues and plants, a lot of it is proteins. Proteins also organize, proteins catalyze reactions, proteins make things happen. And proteins don't do it alone, they do it in cells, and cells don't do things as single cells for big organisms. So it's about doing things at a macro level, at the cellular level, that I think is sort of one of the big things that I like to do, and looking at the mechanics, because in the end you have to make a physical object and things have to move around and take shape. So my interdisciplinary thing, to answer your question, is I love to work with people who do computational modeling. We're actually doing it ourselves a bit now, but I love to talk with engineers and physicists and mathematicians on that side, because I like the explanations to be a bit simpler than the thousands of genes and gene names that, it's important knowledge, but it's sort of at some level, even to very sophisticated perceivers, it's very cognitively hard to take in. You can't, if you've got a list of 10,000 genes, you can't really look at that list in any meaningful way. You've got to find some way of reducing its complexity. And that's where models come in. You sort of simplify, put things in black boxes, and then look at inputs and outputs. So it's that sort of thing. So I think that's part of the interdisciplinarity thing. And it's also more fun because you learn new things when you're dealing with new people.
So I'm going to get into the questions now. So I did, to prepare for this episode, we talked with several people that I've met, at Leicester Square, at the trains, and also parks, and just asked them what they think about Professor Jeremy Greenfield, and any questions they would have for him, just based on the topic. And really, it gives you an idea about public perception about the field, because science is good, but after the science is done, we also have to communicate it and let the public know about it. So I will go into some of the interesting questions that we've got for Prof. Jeremy now. So, do cells ever make mistakes?
All the time. So one of the things, I mean, this is not very close to what I do, but one of the general themes in biology, which is close to what I do, which is that these are very complex machines, if you like. So cells have thousands of components, tens of thousands of components, and there are tens of thousands of cells, actually trillions in the body. So I think what happens is that cells that make mistakes, we know of examples where cells are doing the wrong thing, and funnily enough, there's a paper from Sir John Gurdon, who I mentioned earlier, which he called a community effect, where he deliberately put a cell of the wrong cell type in among others.
Oh, wow.
And it turns out that they change. So there are signals that regulate, that, for example, make this errant cell sort of behave itself. That's called a community effect. There are other examples where cells that have mutations in them will start to become a bit cancerous, and the body has lots of mechanisms for either killing them or otherwise getting rid of them, or stopping them growing, or whatever. So in that sense, they make mistakes.
That's very interesting. Lovely. Why don't our ears grow where our elbows are?
Okay, well, so funnily enough, in some insects, they do hear with their knees. So I mean, it's kind of a funny coincidence. So this is a general question about development. How do things get to be in the right place? And the answer is that stepwise, cells are essentially have a set of instructions. That's all of the genes together, your genes and chromosomes, the DNA, and those instructions are not active all the time. So different sets of instructions get switched on in different cells. So ear genes are genes expressed in the ear. Genes switched on in the ear are different from those in the elbow. And so then the question becomes, how do you switch genes on in one place and not another? And fundamentally, it's the thing I mentioned earlier about morphogen gradients.
Yeah.
So starting from the egg being, as it were, more or less uniform, it gets divided between one side and the other into 2 or 3 domains. And then those domains get subdivided, and those domains get subdivided. And then the cells can move around, because one of the things they can do is move, as well as divide and multiply. And a sort of successive subdivisions, movements, and differentiations between neighbors will put the right organs and structures in the right place.
Well, that's amazing.
And that's it. But it's very stepwise, I suppose, is the sort of short answer.
That's awesome. Will doctors ever grow replacement organs instead of transplanting them?
Well, in some senses, that's done already. But the short answer is yes. So the already part is something that I think probably most people are a bit familiar with, which is burn victims.
Yeah.
Often don't have enough skin to graft to cover their burns, and so what is done nowadays is they get a temporary graft just for their immediate survival. But the doctors, the medics will take some cells and they will actually grow them in a dish.
Yeah.
So they will regrow that skin and then put it back on the patient. And so that's sort of the poster child for essentially for growing the cells. But a lot of what has been in the offing for a rather long time, but now I think it's really happening, is really starting with adult cells of one type, turning them into embryonic cells, reprogramming them into an embryonic state, and then redifferentiating them, redeveloping a particular cell type of interest.
Yeah.
And so the whole interest in stem cells is really is not really about the cells themselves. It's really about how you take an undecided sort of embryonic cell and tell it to become a pancreas cell for a type 1 diabetic patient, or how you could grow a replacement bit of lung or bone. It's not quite there yet in many of the areas where we would like it to be, but there are certainly examples that I think are very promising. I think one of the most exciting, for example, is using embryonic stem cells for the treatment of Parkinson's disease. Yeah. So Parkinson's is a defect in a very, very well-defined population in the brain. And there have now been 3, I think it's phase 1 clinical trials taking embryonic stem cells that have been directed from their stem cell state to that motor neuron state and put back in real people's brains. And it's still phase 1 or phase 1, early phase 2, but it's looking very promising.
Yeah. It's amazing.
Yeah, it's very, very exciting stuff.
If your research succeeds beyond your wildest expectations, how will medicine look different in 50 years?
Well, that way.
Yes,
I mean, that's basically the answer.
Yeah.
So, I mean, we will be able to repair things.
Yeah.
Potentially, I mean, and I think this is probably a rather important point. A lot of what we do in research and in this sort of area is very high tech and very expensive. Yeah. And what you want to do with these very high tech things ultimately is not have them merely for the billionaires. Yeah. I mean, you want to distribute them both within the UK, but of course to the world and to countries that may be less wealthy. And I think that's probably what I would want for this kind of super high-tech stuff, is to do it in such a way that it's very widely distributed to everybody. That's the 50-year horizon. I mean, I'm thinking, for example, one of the things we work on is cleft palate. So a lot of babies are born as relatively common birth defect with either a cleft lip or a cleft palate. Yeah. And we're working on the basic biology of how the palate gets made. Yeah. And were way away from regenerative stuff, or even sort of, well, maybe not so far away from kind of fairly high-tech surgical interventions that
Yeah.
that, as it were, prevent the cleft from happening in the first place.
Yeah.
And so prevention is better than cure. But even some high-tech interventions may ultimately be less expensive than doing surgery after the fact.
Yeah.
So that's kind of would be the ideal on that scale. That
Yeah.
would be my
That would be
the dream.
well, that's amazing. Thank you. Um, just gonna go through them again. When you started your career, what do you think was impossible? What was impossible but now has been achieved? This is probably the last one from the questions.
Okay.
Yeah.
Well, that's actually a hard question. I think that when I started, the whole field of developmental biology was pretty theory based. We didn't know any of the molecules or actual, if you like, the chemistry
Yeah.
of how it worked. The theories all seemed very plausible, but they didn't have any material evidence,
Yeah.
direct material evidence. And now we really do know what those morphogens that I mentioned earlier, what they are. We don't necessarily know every morphogen, but we know the main ones. As it turns out, there are 6 main ones, and each one comes in a bunch of different flavors.
Yeah.
And so you've got this sort of almost like not a periodic table, but you've got a repertoire, a menu, and most things that happen you can at least manipulate with those, even if other ones are involved. And so I think the thing that was impossible was to know all of that, and now we know. I mean, the stuff I do is very basic science, so it's really more about the basic biology, the basic wonder. Like astronomy is not of itself directly useful anymore. I mean, we don't use it for navigation, for example, but we know that the whole process of knowing about the cosmos is a foundation of a lot of other science, and a foundation of some technologies as well. And I think that that's what I like to do. I'm if one sort of one of the metaphors I think of in science is like mining. So there are people who do, the equivalent of strip mining, right? They've got technology where they can, scoop up huge, vast amounts of data, and it can be valuable in that way.
Yeah.
And, there are the sort of deep mine shaft builders, and I see myself as sort of the lone prospector with the pony and the pickaxe, because I like to be right there because I love the exploration aspects of it.
That's amazing.
So that's probably what the impossible is knowing the next thing, if you like.
Oh wow, that's amazing. So it's just amazing that going from theory to the material of chemistry of it, actualizing it. It's quite a long process. It might take a lifetime for a lot of fields. So what sort of advice have you got for aspirational scientists, who've probably just finished their first degree, or they have finished their PhDs and they're looking for a long-term postdoc or a short-term contract? What sort of one or two can they learn from you?
Obviously work hard.
Yeah.
Don't give up. You have to do something that you care about. That's probably for life as well, not just science. But you need something that keeps you going. And I think most people who are scientists who get to that point, are quite exacting, quite demanding of themselves. And I just wonder, sort of as a word of encouragement, you're probably better than you think you are, and just keep that in mind and stand up for yourself.
Yeah, that's amazing. So who's played that role for you, sort of inspired you over time and just kept you going?
Oh, um, I think I would sort of have sort of everybody. I mean, a lot of scientists. I mean, a
Mm.
lot of, you almost all my colleagues. I mean, almost all of the people in the profession. I mean, it's one of the great things about being in science is and working in science is you're surrounded by a lot of smart people, a lot of
Yeah,
brilliant people, and I'm inspired by uh, not necessarily even people more senior than me. I mean, you find a student who's just is really good and is really engaged and excited, and that's actually very motivating.
yeah.
So I tend not to focus on one particular role model. I mean, I have to thank Jim Smith, for example. I have to thank my PhD supervisor, my mother, all of that. Um, my father, all of that. I um, I think you have to gather inspiration from everywhere you can.
Yeah, it's amazing. I think we're coming to the last question now. It's just
Okay,
been thank you for having time out of a busy schedule to do this. But if we had this interview again or this chat again in 50 years, what do you think hopefully would be different for you?
different. Well, I'm old enough that I know I probably won't be around in 50. Well, actually, being around in 50 years is
Yeah,
probably
it's
the
probably the
big
big
different
achievement.
thing. Although, you want eternal youth, not eternal life. So hopefully, I'll be, even younger looking and fitter and all that stuff. Um, that's actually a really hard question. So, can you run it by me
Yeah,
again?
so if we had this chat
Conversation.
conversation again in not okay 20 years 30 years from now,
Okay, all
and
right. Giving
how
my life expectancy more realistic.
yeah, how do you what would be different about hopefully about your field, about your approach to science, or your the discoveries and innovation in developmental biology?
Um, I think actually realizing the vision of regenerative medicine, which has been an idea that's been around in the field for quite a long time. Well, I have to say that the stuff I was doing 30 years ago was definitely not for a technological aim. It was like, I want to know how this works.
Yeah,
But the real applications of this, um, to be able to do repairs of birth defects, which are still a very serious cause of death. I mean,
yeah,
they're bigger than many other of the headline causes of death and perinatal death that you might know about. To actually be able to have applied those things and made a lot of the promise, which is still not realized, for it to be realized.
yeah,
And I think 20 to 30 years is probably about the right timeline for that really to come to fruition. We're not there yet.
yeah.
We can see the outlines of the route, but it's not been realized yet.
Yeah. Thank you. Well, thank you so much, Prof. Jeremy Green, for being in today's episode.
Pleasure.
Thank you for listening in, and I hope you click subscribe, share to this video, whatever you see it on YouTube, on LinkedIn, on TikTok, and Instagram, and let's just keep spreading science and keep inspiring more people, younger talent, to also join the research field, and also just get to see the behind all the amazing discoveries that we are seeing in research are people with lives and amazing stories to share, and it's just wonderful. Thank you so much. Thank you.
You're welcome.